In my talk today, I would like to cover 4 main points. Firstly, talk about the microbiota as the reservoir for bacteria, resistant bacteria, and resistant genes. And then what are the consequences of this colonization? And at the end, briefly touch on how we can actually modulate microbiota to prevent this colonization. So what are the multi-drug resistant bacteria and which ones are the nastiest? Multi-drug resistance is defined as resistance to 3 different antibiotic classes. And the World Health Organization has made a list of those pathogens that should be the priority for the research because of the severity of the disease, the difficulties in treatment, and the overall impact of the disease. And they are listed here in the middle. And the resistances that we are mostly fearing are those against the different beta-lactams, which are mainly carbapenems or cephalosporins. And strains that do have these resistances very often tend to collect other resistances as well. So they are also multi-drug resistance, and the carbapenem resistance or extended spectrum beta-lactamases are sort of the marker for multi-drug resistance. So all those main bad guys that are listed here are found on our different microbiotas. So this is sometimes their natural reservoir, or they can live in microbiota. But saying this, it's also true they are present, but if everything is normal, they're present in very low numbers. Why is this so? This is so because the microbiota, any microbiota, has, among other roles, a very important role of colonization resistance. Colonization resistance is something that basically, excuse me, yes. Could you stop? Okay. Colonization resistance is taking care for stability of each microbiota, because we are constantly encountering bacteria, but not every bacteria can become a part of our microbiota. And colonization resistance is very important for preventing the pathogens to colonize us. The mechanisms of colonization resistance are very diverse and various. They include the indirect mechanisms that goes via immune system, and direct mechanisms. And direct mechanisms would include 2 main parts, either microbe-microbe interactions, so bacteria can produce some bactericides or other compounds, or other side would be based on metabolites, either competition for metabolites or not providing metabolites, or taking the specific metabolites out of the ecosystem. And the consequence of this very diverse and different colonization resistance mechanisms is actually the fact that for each specific pathogens, we do have specific combinations of our protective bacteria. So we do have specific consortia that protect us, and we will come to this again later. Now, just to give you an example how the indirect mechanisms would work, This is true for enterococci. If everything is okay, our gut microbiota stimulates the Paneth cells to produce the antibacterial peptides called Reg3. If we take antibiotics, then we destroy these commensals and we destroy the signal for the Paneth cells, and the peptides are not produced anymore, and Enterococcus can start to grow and regrow in our microbiota. So our colonization with multi-drug resistance organisms or multi-drug resistance bacteria is the consequence of two factors. One is dysbiosis in the microbiota, and from now on I will focus on gut microbiota, and the second one is exposure. What is dysbiosis? Dysbiosis is the state of unhealthy microbiota that cannot provide all the functions. What are the factors that are enhancing the dysbiosis? There are several. There can be connected with our lifestyle. There can be influenced by disease, and disease means inflammation, and inflammation in the gut means that the oxygen levels will go up, and oxygen levels will destroy anaerobes, and then the Enterobacteriaceae can take over and start to prevail in the ecosystem. And the third thing that greatly influences the dysbiosis is everything that we eat, either as a diet or unintentionally included in the diet. And there is a very nice review published in a couple of years ago, summarizing, as you can see here below, summarizing hundreds of components that we are actually ingesting and slowly affect or destroy our microbiota. So we somehow automatically always connect gut dysbiosis with antibiotics, and this is true. Antibiotics are a very important driver of dysbiosis, but here I'm showing you the preliminary results of our study where we are following 9 healthy volunteers and how their microbiota changes during the time. And you can see that each of us is actually circling around some sort of steady state, and even if we are healthy, we do from time to time come into some sort of dysregulated state that can be very short term, but repeatedly we do have these states. So this is true if we are healthy. Patients in the hospitals, they tend to have more disbalanced microbiota, and this is another one of our studies where we have looked at the patients at one single ward and compared them with the healthy cohort. And here you see that basically our patients, their gut microbiota could be distributed into 3 main clusters. Important things here are 2. First, the clusters are not in any way correlated with the diagnosis. Each cluster contains all different gastrointestinal diagnoses. And the second one is that we saw these 3 clusters, and 2 of them were really, they really had specific microbial signatures, which means dysbiotic gut opens up the niche on one side for gram-negatives, mainly Enterobacteriaceae, and the other type of dysbiosis open up the niche for Enterococcus. And as I showed you before, these are 2 groups with the main bad guys for the multi-drug resistant bacteria. So once the niche is opened, then it depends on the exposure. If we are exposed to these bacteria, if they are susceptible or if they are resistant. And the main ways of exposure are listed here. A lot of bacteria we get with the social contacts or with the food or from the environment. And hospital environment is in particular dangerous because people will get there, will leave their multi-drug resistance bacteria in the environment. New patients will come and will pick this bacteria up. So in general, in human population, the colonization with multi-drug resistance bacteria is increasing through time, and the bad news is that this increase is higher in the community in comparison to the hospitals. But in the community, if we are healthy, our carriage rate is occasional and transient, and it's very short term, couple of days. This is well illustrated in one paper from the group of the medical professionals that were traveling to Laos, and each and every one of them was colonized with at least one multi-drug resistant bacteria, but only for a couple of days, okay? But after hospitalization or during hospitalization, the colonization becomes much longer. And once resistant bacteria comes to our gut, they tend to leave their genes in our gut commensals. And looking at the large number of publicly available metagenomes, we could find, not we, others could describe that approximately 15 resistance genes per microbiome were found. Different sites had different resistances, but obviously the mouth and the gut were the hotspots. Our commensals are basically the reservoirs. They're collecting the resistance genes from the pathogens. So pathogen will come, would leave the resistance genes in our commensal pool, and the pathogen will go. And the new pathogen will come, maybe the susceptible one, and will take the resistance genes and will become resistant bacteria. This horizontal gene transfer is even higher in the presence of antibiotics and is higher in the presence of inflammation. In every country, a certain proportion of people would have the resistance genes in the gut microbiota, and diversity of resistance genes would be different in different countries, and the diversity looks very low in China. But then if you look the abundance, so the proportion of the population with the resistance genes, you can see that this is quite high. And another bad news is that situation might be even worse, because we do a lot of studies on fecal samples, but because fecal samples are sort of easy to get, but the gut luminal sites and the mucosal sites are difficult to sample. But if you do this, what the study finds is that there is even more resistant organisms and more resistant genes in the gut when we compare those non-fecal samples with the fecal samples. Okay, so what is the consequence of this colonization? On one side, people that are colonized, they are spreaders and they are source of infection with multi-drug resistant bacteria. On the other side, they're in danger because those that are colonized have higher risk to develop infection. because if they are colonized, they are very likely to have dysbiosis. Dysbiosis means leaky gut. Leaky gut means that bacteria will travel to extraintestinal sites. So these two are quite logical things. The third consequence is perhaps a little bit more surprising, but maybe not. The third consequence is that all other extraintestinal infections will be more severe or will be worse. And this was, for instance, shown in one mouse model where colonization with carbapenem producing Klebsiella was associated with a worse infection in the lung with Pseudomonas aeruginosa. Why is this so? This is so because of the gut-lung axis and influences on the immunity as such. So if we know that colonization and gut microbiota is so important, then it's quite logical to start developing the approaches to influence gut microbiota or protect gut microbiota, and thereby to minimize the colonization with multi-drug resistant bacteria. There are several ways how it could do this, and two of them are now heavily researched. The microbiota protection was somehow on the radar, and there were some products developed, for instance, a specific charcoal that was active or released in the colon and could then absorb the remnants of antibiotics. And what is also developed and in clinical trials are oral beta-lactamases. So you actually take the pill with beta-lactamases, something that we don't want if bacteria have them. But if we take these enzymes and these enzymes would get to the gut, they will again destroy the beta-lactams in the gut, and in this way protect our gut microbiota. But the main two ways that are now in development are use of probiotics or prebiotics and the fecal transplantation. Probiotics seems to be working a little bit better with gram-positives than in gram-negatives. In some occasions, probiotic could even increase the level of resistance genes, and probiotics are effective only in permissive individuals. And FMT is now very hot topics, and it's in clinical trials for several health conditions. And you can see very high on the list is also prevention of colonization with multi-drug resistance microorganisms. So in summary, gut microbiota is important reservoirs for bacteria, for genes. Colonization is the summary of dysbiosis and exposure, and we should try to decrease both of them. And interestingly, the models show that increasing only one or only the other has poorer effects than if we do both at the same time. And lastly, we are now seeing developments of new microbiota-targeted approaches to decrease colonization and infection burden. And with this, I would like to close and thank to my group and to you for attention. Thank you very much.